Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be seeing CUSAT CAT Biotechnology question paper, the exam which was conducted in the year 2021. So coming to the first question, the term cell is not used to describe the following living systems. So algae, bacteria, virus and fungi. The answer is virus, that is option C, because viruses are not true cells. They have a simple coat of protein only and they do not contain any cell organelles. Then viruses are alive only when they are inside their host cell. Otherwise they behave as dead cells when outside the host cell. Hence and and they are also an exception to cell theory. So the term cell is not used to describe virus. Then next one. Developing GM brinjal in India had the objective of. So genetically modified brinjal that are found in India are insect resistant or pest resistant. This is because of the presence of the Bt gene. The gene is responsible for producing toxin that kill the insects. And Bt brinjal, it gives resistance against lepidopteran insects, that is fruit borer and shoot, brinjal fruit borer and brinjal shoot borer. So the answer is option C. Then next one. For which of the following health conditions was gene therapy tried first? So what is gene therapy? Gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of the disease causing mutation. Then the first patient to be treated with gene therapy was a 4-year-old girl, and it was conducted at the NIH Clinical Center in the year 1990. She had congenital disease called adenosine deaminase deficiency, which severely affects immunity and the ability to fight infections. So the first gene therapy was for the enzyme ADA deficiency, that is adenosine deaminase deficiency. The excised part of plant and animal used for in vitro culture is called as the explant. Answer is option A. Then next one. The insecticidal protein synthesized by Bacillus thuringiensis brings about its action. So Bt strains, they synthesize Cry proteins as well as CYT toxins, that is crystal toxins and cytolytic toxins. And this is produced at the onset of sporulation. And once ingested by insects, these crystals are solubilized in the midgut. The toxins are proteolytically activated by the midgut proteases and bind to specific receptors located in the insect cell membrane, leading to cell disruption and insect death. So the answer here is by binding and destroying midgut epithelial cells in insects. Answer is option C. It is not, you may get confused with option A also, that is being toxic during insect ingestion. That is while the insect is intaking the toxin, it is getting toxic effect. But actually it is it is only after it gets enters into the midgut, that is the intestine, midgut epithelial cells in the insects, that it will bring about its action, the Cry gene. Then EDTA is used for in vitro subculturing of animal cells So subculturing is also referred to as passaging in the case of animal cells, and is the removal from the old medium and transfer of cells to the fresh medium. And this will enable for further propagation of the cell line or cell strain. Then subculturing also produces more homogeneous cell lines and avoids the senescence associated with prolonged high cell density. Then splitting cells involves transferring a small number of cells into each new vessel. Now if you have adherent cell cultures, so they have to be detached from the surface of the tissue culture flasks or dishes using proteins. So proteins secreted by the cells form a tight bridge between the cells and the surface. This is what happens in adherent cell cultures, because in adherent cell cultures the protein secreted by the cells They form tight bridge between the cell and the surface to which they are adherent. So you need you need other proteins such as trypsin for detaching these adherent cell cultures so that they could be subcultured. So for this you will use a mixture of trypsin and EDTA. Now trypsin and EDTA mixture is used to break proteins at specific places. Trypsin is either protein degrading or proteolytic, and it hydrolyzes pepsin digested peptides by hydrolysis of the peptide bonds. Then what does EDTA do? It will sequester certain metal ions that can inhibit trypsin activity and thus enhance the efficiency of trypsin. So the answer here is option A, that is chelates the metal ions that participate in cell adhesion. Then next one, expression systems available for large-scale recombinant protein production in mammalian systems include. So for mammalian systems it is baculovirus. Answer is option A. Agrobacterium is for plants and T4 phage is for bacteria. Next one, which type of culture can be used to get virus-free plants? So it is Meristem culture. This question was asked not only for CUSAT but also for other universities also. They're like Hyderabad University, Pondicherry University, they also have asked all these questions. I have answered most of the questions of Hyderabad University and Pondicherry University. So you can refer them for gaining more experience while answering multiple choice questions. So why Meristem culture is used for getting virus-free plants? So meristematic tissue, it is generally free of viruses because it is actively dividing cells, and this reduces the differentiation of vascular tissues. So that is why we use meristematic stem culture to obtain virus-free plants. Then titrated antibodies are used in. So what is titration? Titration is the process of identifying the correct concentration of antibody to use for any given assay. Now it is also used to ensure the antibody performs within acceptable parameters. The non-specific binding of antibody is also eliminated by optimizing the amount and concentration of the antibody. So the titrated antibodies, they are used in the case of radioimmunoassay or RIA. Then next one, second line of defense in providing immunity includes. So the second line of defense, it is non-specific resistance that destroys invaders in a generalized way without targeting any specific cells. So these include the phagocytic cells or the macrophages and destroy all microbes that pass into body tissues. So answer is option A, that is macrophages. Then next one, urea denatures protein by. So urea denatures protein by weakening the intermolecular bonds as well as changing the hydrodynamics of the solvent. So the answer is option D, that is both A and B. Then 5S, 16S, 23S RNA molecules are seen in. So they are usually seen in prokaryotic rRNA. Answer is option C. Then first genetically engineered model plant was. So the first genetically engineered crop plant was tobacco, and it was reported in the year 1983. So answer is antibiotic resistant tobacco. The next one, the gene coding opine synthesizing enzymes are present in. So opines are used by bacterium as an important source of energy, that is carbon and nitrogen source. Then these are usually low molecular weight compounds and found in plant crown gall tumors, and it is produced by the bacterium belonging to genus Agrobacterium tumefaciens and Agrobacterium rhizogenus. And opine biosynthesis is catalyzed by specific enzymes encoded by genes contained in the small segment of tDNA, which is a part of Ti plasmid in the case of Agrobacterium tumefaciens and Ri plasmid in the case of Agrobacterium rhizogenus. So the answer here is Ti and R plasmid. They have not given Ri. It varies from notation may vary from textbook to textbook. So the answer here is option C. The next one. Clino fragment of DNA polymerase 1 retains. So what is clino fragment? It is a large protein fragment produced when DNA polymerase 1 from E. coli is enzymatically cleaved by the protease subtilisine, and it retains 5' 3' polymerase activity and 3' 5' exonuclease activity, but it does not have 5' 3' exonuclease activity. So what they have asked here is it retains, clino fragment of DNA polymerase 1 retains, it retains 5' 3' polymerase activity. It is not 3' to 5' polymerase activity, it is 3' to 5' exonuclease activity. And option C, that is 5' to 3' exonuclease activity, that is actually lost by the clino fragment. It is not present in clino fragment. Then next one. Fermentation system in which fresh medium is delivered at a constant rate and cells and spent medium overflow at the same rate is. So it is in the case of chemostat. Answer is option A. The next one. The key regulatory enzyme of TCA cycle among the following. It is isocitrate dehydrogenase. Answer is option C. This enzyme regulates the speed at which the citrate isomer, that is isocitrate, loses a carbon to form 5-carbon molecule, that is alpha ketoglutarate. So isocitrate is converted to alpha ketoglutarate by losing one carbon atom. So, and it is carried out by the enzyme isocitrate dehydrogenase. So that is the regulatory enzyme in TCA cycle from these options. Then next one. Among the following, which is used to break the vortex formation in the fermentation vessel? Now that is baffle. Answer is option A. They are called as baffles. Now the important function of baffles is to break the vortex formed during agitation process by the impellers. So if this vortex is not broken, then what will happen is the fermentation media will spill out of the fermenter, and this may result in contamination as well as can lead to other different problems also. So it is important to break this vortex formation, and it is caused by impellers. Okay. And for this you use baffles. Baffles act as barriers which break the vortex. The next one. Beta oxidation in eukaryotes take place in. So beta oxidation, it take place in the mitochondrion. Answer is option D. Then next one. Prokaryotic translation initiation requires energy as. So it requires energy in the form of GTP. Answer is option B. Hydrophobic component of electron transport chain include. So the hydrophobic component of electron transport chain is ubiquinone. Answer is option C. Then the rotor, which avoids Wall effects during centrifugation. Now Wall effect is usually encountered in swinging bucket and fixed angle rotors, and this can be minimized by using zonal rotors. So answer is option D. The next one. The relationship between Beer-Lambert law is. So Beer-Lambert law is given by absorption is equal to molar efficiency coefficient into concentration into path length. So energy, absorption and concentration, that is A is equal to epsilon CL. So absorption is A, epsilon is molar efficiency coefficient, C is the concentration and L is the path length. So energy absorption is equal to epsilon CL. So answer is option A, that is energy absorption and concentration. Then Salk's vaccine for polio was manufactured first using. So for this, HeLa cells were used. HeLa cells were used by Jonas Salk to test first polio vaccine in the 1950s. They were observed to be easily infected by poliovirus, causing infected cells to die. This made HeLa cells highly desirable for polio vaccine testing since results could be easily obtained. So answer is HeLa cells. Then vesicular arbuscular mycorrhiza is formed by the symbiotic association between fungi and angiosperm roots. Answer is option A. The fungus colonizes the root cortex, forming a mycelial network and characteristic vesicles and arbuscules. So answer is option A. Then next one. Chinchilla is a species of. It is a species of rodent. Answer is option B. Then total number of bones in neonatal baby is. So it is in the case of neonatal baby it is 300. And these 300 they will eventually fuse to form 206 bones in the case of adults. So answer is option C. Then which organ secretes trypsin? So it is the pancreas that secrete trypsin. Answer is option C. Trypsin is produced by pancreas in an inactive form called trypsinogen. And trypsinogen is cleaved to its active form trypsin by enteropeptidase, which is found in intestinal mucosa. So the answer is option C, that is pancreas. Then next one. Deficiency of protein can lead to. So deficiency of protein is can lead to kwashiorkor. Option beriberi is due to deficiency of vitamin B1, that is thiamine. Scurvy is for vitamin C, and osteoporosis it is a lifelong lack of calcium. So answer here is option D. Then which is anti-hemorrhagic vitamin is essential for coagulation of blood? So that is vitamin K. Answer is option D. The digestive enzyme ptyalin is a salivary amylase. Answer is option C. These are all direct questions, that's why I am not explaining much. Next one. Exchange of bicarbonate and the chloride across the membrane of red blood cells is. Now this process it is called as chloride shift. Answer is option A. Then sphygmomanometer is used to measure. It is used to measure blood pressure. Answer is option A. Tube feet of locomotory organs is. Tube feet are locomotory organs in. Okay, sorry. So they are locomotory organs in the case of echinodermata. Answer is option C. Then torpedology is the study of. It is the study of sharks. Answer is option B. The receptors in fish responding to water currents are. So they are called as the rheoreceptors. Answer is option B. Then chills and rigors in malaria is caused by. It is caused by hemozoin. Now hemozoin it is referred to as malarial pigment. And its formation is important for the survival of the parasite in the host body and discharges parasitic waste that are known to cause fever and chills in malarial patients. So the answer is hemozoin. Next one. Which organelle in amoeba regulates osmotic pressure in the cell? So it is the vacuole. Answer is option D. Then movement of paramecium in an electric field is. It is called as galvanotaxis. Answer is option A. Then tears are produced from. So tears are produced from lacrimal glands. Answer is option C. Then ink glands in sepia produce. They produce what is called as melanin. Answer is option B. Galacturonic acid is the building block of. So it is the building block of pectin. Answer is option C. And pectin is a polysaccharide based polymer in which the backbone consists of galacturonic acid and rhamnose with side chains composed of arabinose, fucose, galactose and xylulose. Xylose. Then the water conducting element in xylem is. Companion cells, bars, sieve tubes is not the answer, it is tracheids. Answer is option A. Then next one. Elements that are responsible for transporting sugar throughout the plant are. So they are sieve tubes, that is phloem, part of the elements of phloem. Okay, so the answer is option B. Which plant is mainly used for biodiesel production? This is a very common question for most of the entrance exam. Answer is option C, that is Jatropha. Which elements deficiency in soil support the growth of insectivorous plants? That is nitrogen. This is also a common question for many MSc entrance exams. Answer is option C. Then, actively dividing cells found between xylem and phloem tissues in plants it is called as the cambium. Answer is C. Then the theory, active site of an enzyme is precisely shaped to hold the substrate, was proposed by. So we will see one by one. The induced fit hypothesis is a model depicting the enzyme-substrate interaction. The substrate bound to the active site of an enzyme undergo changes to attain an optimal fit. So that is the case of induced fit hypothesis. Then substrate strain theory. Substrate binds to the preformed active site of the enzyme and induces a strain to the substrate. The strained substrate leads to the formation of product. So what is left out? Lock and key theory. So the active site of an enzyme is precisely shaped to hold the substrate, just like a lock, just like each key for a specific lock. So answer is option C, that is lock and key theory. The next one. Oxygen carrier found in nitrogen fixing root nodules of leguminous plants is. So it is called as the leg hemoglobin. Answer is option C. This is also a repeated question for many MSc entrance exams. Then next one. Heterotrophic plants with no chlorophyll include. So what are saprophytes? So saprophyte is a plant that does not have chlorophyll, and they obtain its food from the dead matter similar to bacterium and fungi. So answer is saprophytes. Answer is A. Then which is a growth inhibitor? So abscisic acid is a growth inhibitor. Now it inhibits germination and seedling growth. It enhances degradative changes such as ripening and senescence, especially in the case of senescence of leaves. The next one, L-carnitine occurs in. So L-carnitine, it is found in the mitochondria. Now L-carnitine, it is a naturally occurring substance required in mammalian energy metabolism that function by facilitating long chain fatty acid entry into cellular mitochondria, thereby delivering substrate for oxidation and subsequent energy production. So that is L-carnitine. So answer is option B, that is mitochondria. The next one. Light independent reaction in photosynthesis is responsible for reducing power in NADPH, oxygen evolution, yes, ATP generation, yes. So answer is all of the above. Then first stable compound formed in both C4 and CAM pathways in plants. So in both the plants, PEP carboxylase enzyme act on carbon dioxide acceptor and forms oxaloacetate, which is the first stable compound. So answer is option B. Then amphibians of the plant kingdom include. So they include the bryophytes. And why are they called so? Because bryophyte plants require water for reproduction and they are called as amphibians of the plant kingdom. So answer is option A. Next one. The plant hormone produced during chilling is vernalin. Answer is option D. Then germination of seed below the ground is termed as, it is termed as hypogeal. Answer is option A. And epigeal germination refers to the germination of the plant that take place above the ground, while hypogeal germination refers to germination of a plant that take place below the ground. Okay. The next one. Plant variety produced by selective breeding is. So what is a hybrid? We'll see option B first. Hybrid is a crossbred plant that is the result of cross-pollinating two or more unrelated plant species. That is called as a hybrid. Then what is a pure line? Option D. Pure line is the progeny of a single self-fertilized homozygous plant. It is totally pure line. Okay. Because it is from a single, self-fertilized or self-pollinated homozygous plant. That is called as a pure line. And what are mutants? The plants developed by the process of exposing seeds to chemicals, radiations, or enzymes in order to generate plants with desirable traits to be bred with other cultivars are called as mutants. So plant variety produced by selective breeding is called as a cultivar. Answer is option A. And next, algal bloom leads to. So algal bloom is rapid increase or accumulation in the population of algae in fresh waters or marine water system, and it results are the result of, and these algal blooms are the result of nutrients such as fertilizer or runoff nitrogen or phosphorus entering the water environment and causing excessive algal growth. So the answer here is option A is oxygen depletion from the aquatic ecosystem. Yes, that is the result of algal bloom. Then secreting toxins in the aquatic ecosystem. That is also one of the result of algal bloom. So answer is option C, that is both A and B. Then anaerobic digestion of organic waste generates. It generates methane. Answer is option A. No two electrons can have all the four quantum numbers to be the same. Is the basis of. So it is the basis of Pauli's exclusion principle. Answer is option C. The next one. Penetrating power and ionizing capacity of alpha, beta, gamma rays are. So gamma radiation is the most penetrating of the three radiations, and beta radiation is more penetrating than alpha radiation. So option B, if you see alpha less than beta less than gamma, that is correct. And ionization in case of gamma radiation, it is high for, ionization in the case of alpha radiation, it is very high. Intermediate for beta and very low for gamma. So option B again, if you see alpha greater than beta greater than gamma. So answer is option B. Then next one. Fluid's internal resistance and friction is described by. This is described by viscosity. Answer is option A. The next one. Tears of wine phenomenon is due to. Now tears of wine are droplets or streaks of water that form on the inside of a wine glass as you move the wine around in the glass. That is called as tears of wine. Now this phenomenon, it is due to differing surface tensions of water and ethanol. Answer is option C.
So coming to the next question. How long will it take for 40 gram iodine 131 half-life of 8.040 days to decay to 1 by 100 its original mass? So first we have to determine the number of half-lives. After n half-life, the initial reactant concentration is reduced by half raised to n. Half raised to n. So half raised to n, they have given it as 1 by 100, that is 0.01. Taking logarithm on both the sides, n log 5 is equal to log 0.01. N is equal to minus 2 by minus 0.301, that is 6.64 half-lives. Now time elapsed is equal to n into t half. So 6.64 into 8040 days will be 53.4 days.
Okay, then next one. Electronegativity of an atom can be measured by. Now what is electronegativity? The tendency of an atom in a molecule to attract the shared pair of electrons towards itself is known as electronegativity. And this can be measured by using AFM and SEM. AFM is atomic force microscopy. So answer is option C, that is both A and B. Now, How many enolic forms can arise by tautomerism in the below given ketone? So you will get 2 enolic forms. Answer is option D.
Now coming to the next question. What is the molarity of 5 gram of NaOH in 750 ml of solution? So we have got the formula for molarity, that is 1000 into Wb by Mb into V, where Wb is the mass of the NaOH, Mb is the molecular weight of NaOH, and V is the volume in ml. So 1000 into 5 divided by 40, that is the molecular mass of NaOH, into 750, you will get 0.1666. So you can approximate it to 0.167 molar.